Hey everyone, ready for a deep dive into DNA?
I know, I am.
We're cracking open DNA, the code of life.
By Mr. Chi?
Yeah, by Mr. Chi. It's
Really great book.
yeah, it is tons of pictures and diagrams, really accessible, but we're going to try and pull out some of the most interesting stuff.
Definitely. And we'll go beyond just like the textbook stuff,
Yeah.
how it actually works.
Yeah, and how it affects us day to day.
Impacts our lives for sure.
So one thing that I think is really cool is that okay, you've got your cells right.
Right.
And in the center of the cell is the nucleus.
Right.
But did you know that a small percentage of your DNA is actually not even in the nucleus?
Yeah, that's right. It's pretty wild.
So we'll get to that later. But first, I think we got to back up
Okay.
and kind of talk about what DNA even is.
Okay, sounds good.
So, everyone's heard of DNA, right?
Yeah.
This double helix.
Twisted ladder, yeah.
But what does that actually mean?
Right, so it's literally like a ladder that's been twisted, and the sides of that ladder are made up of sugar and phosphate molecules, and then those rungs in between are the nitrogenous bases.
So it's like a language with just 4 letters.
Exactly, yeah, just 4 letters. Adenine, thymine, cytosine, and guanine.
A, T, C, and G.
Right.
But they pair up.
They always pair up in a very specific way. A with T and C with G.
So the order of these letters is what makes up our genetic code.
It determines everything about us.
And that double helix shape, that twisted ladder shape, that helps it copy itself, right?
Yeah, exactly. When it comes time for the cell to divide and make a copy of itself, that ladder unzips down the middle, and each half can serve as a template to build a new matching strand.
Oh, so that's why they call it a code.
Yeah.
It's a set of instructions.
Exactly. Blueprint, a code.
That can be copied and passed on.
Exactly.
Okay, but DNA does more than just pass down traits, right?
Right, for sure.
It's active all the time, right?
Constantly, yeah. So DNA controls cell function. It regulates which genes are turned on and off.
Like a conductor of an orchestra?
Yeah, a complex symphony of activity within each cell constantly going.
So it's not just a static blueprint. It's like an active player.
Active, yeah.
Okay, so how does DNA direct the creation of, literally everything?
Right.
It doesn't build stuff directly, does it?
That's where we have to bring in RNA.
Oh, yeah, RNA.
It's the intermediary. That's what carries the instructions from DNA out into the cell to actually build the proteins.
So DNA is like the architect, and RNA is like the construction crew.
Love that analogy, yeah.
Okay, so proteins are the building blocks of us.
Everything.
So RNA is crucial for translating that code into something tangible.
Absolutely.
Now, remember learning about different types of RNA, like mRNA, tRNA, rRNA?
Yeah, there are a few key players.
How do those all fit in?
Okay, so mRNA, messenger RNA, think of it like a blueprint copy.
Okay.
It's created in the nucleus using DNA as a template, and it carries the code for a specific protein out to the ribosomes. Those are like the protein building factories.
Okay.
At the cell.
So DNA stays safe in the nucleus?
Safe and sound.
And mRNA is like the mobile instructions.
Yeah, exactly. Then you got RRA, ribosomal RNA, which actually makes up the structure of the ribosome.
Okay.
So that's like the factory building itself.
Gotcha.
And then last but not least, tRNA, transfer RNA.
Uh-huh.
And that's like a delivery truck.
Okay.
It brings the specific amino acids, which are the building blocks of proteins, to the ribosome based on the instructions on that mRNA.
Okay, this is all this is crazy, right? It's so intricate.
Yeah, it is.
What happens when there's mistakes?
Well, that's when you get mutations.
Oh, yeah.
Sometimes during DNA replication, an error can slip in, and that changes the sequence of those base pairs.
Like a typo.
Exactly, a typo.
In the DNA.
Yeah, and these typos can have a lot of different effects.
Okay.
Some are silent. They don't change the protein that's built at all. Some cause minor changes, and some can have really dramatic effects on protein structure and function.
It's like those dramatic changes, those could be linked to diseases and stuff, right?
Potentially, yeah.
Or even adaptations, how we evolved.
Exactly, yeah. It's like a game of chance.
Uh-huh.
Some mutations might give an organism an advantage
Right.
in a certain environment, and some might be detrimental.
Yeah, so it's mutations are basically experiments.
In a way, yeah.
In the genetic code.
That's a good way to think about it.
Sometimes they're good, sometimes they're bad, sometimes they do nothing.
Exactly.
This all makes me think of DNA profiling, too, which is it seems like sci-fi, but it's real.
It is real, yeah.
So, how does that work?
Okay, so basically, scientists can analyze specific regions of DNA that are really highly variable between individuals.
Uh-huh.
And these variations create unique patterns that can be used for identification. like a fingerprint.
So they're not looking at like the whole DNA sequence?
Not the whole thing, no.
Yeah. Just like these specific markers.
Yeah, specific markers, yeah.
Okay.
Like you said, like a fingerprint.
Yeah.
You don't need to see the whole hand, just the swirls and the ridges.
That makes sense.
Yeah.
And this is used for so many things, right?
So many things. Forensics, paternity testing, tracing ancestry.
Oh, yeah.
Identifying predispositions to certain diseases. It's incredible.
Like all this information is hidden within our DNA.
It is. It's amazing.
So I think this is a good place to pause for now.
Okay.
But I am really eager to kind of get into the actual step-by-step process of how proteins are built. Like we know the ingredients,
Right.
but we don't know the recipe yet.
We need the recipe. Yeah, we'll get there.
All right, so we'll pick that up in the next part of this deep dive.
All right, so let's get into that recipe.
Yeah, let's do it.
So 2 main acts in this cellular drama: transcription and translation.
Okay, so transcription first.
That's first, yeah. It all happens in the nucleus. That's where we make that mRNA blueprint copy.
Right, like copying the architect's plans before heading out to the construction site.
Perfect analogy.
So how does it actually work?
Okay, so the DNA molecule, it unwinds, it separates those 2 strands, and one of those strands acts as a template for building the mRNA molecule.
So it's like using one side of the ladder to make a new one.
Exactly.
Okay.
And then free-floating RNA nucleotides, they pair up with their complementary bases on that DNA template.
Okay.
Remember, RNA uses uracil, U, instead of thymine, T.
Right, right.
So A pairs with U, and C still pairs with G.
So that base pairing, it's like it makes sure the information's copied correctly.
Exactly. Very precise.
That's so cool.
And once the whole gene's been transcribed, that mRNA molecule, it detaches from the DNA and it heads out of the nucleus through a nuclear pore.
Like a little courier carrying instructions.
It's on its way.
So now it's in the cytoplasm, ready for translation.
That's right. Translation's where that mRNA message is actually decoded and used to assemble the protein.
Okay, and this is all happening at the ribosome, right?
Ribosome is the place to be.
And this is where like the tRNA comes in with the amino acids.
Exactly. Ribosome binds to the mRNA and starts reading the code.
Uh-huh.
And every group of 3 bases on that mRNA, that's called a codon, it specifies a particular amino acid.
Like a 3-letter word.
Exactly. 3-letter word.
That tells the ribosome what to grab.
Exactly.
Yeah.
And this is where tRNA comes in.
Yeah.
Each tRNA molecule, it carries a specific amino acid, and it has what's called an anticodon, which is a 3-base sequence that matches up with a codon on the mRNA.
Like a lock and key?
Perfect. Lock and key system. Only the tRNA with the matching anticodon can deliver its amino acid.
Wow.
And the ribosome's like the matchmaker, bringing the mRNA codon and the tRNA and anticodon together.
Wow.
And as the ribosome moves along the mRNA, it reads each codon. The corresponding tRNAs bring in their amino acids.
And they're linked together to make the protein.
Like beads on a string, forming that polypeptide chain. And it keeps growing till the ribosome hits a stop codon on the mRNA, and that signals the end of the protein.
So this whole crazy process, it's happening like all the time.
All the time, in every cell.
In our bodies.
Yeah, pretty amazing.
What about those mutations, the typos?
Right, so those typos in the DNA code, if a mutation happens within a gene, it can change the mRNA codon during transcription.
So instead of the right instructions, it might get a garbled message.
Exactly, garbled message.
And then the wrong amino acid gets added.
Exactly. And sometimes it's no big deal, but sometimes it can really mess things up.
Like swapping an ingredient in a recipe.
Yeah, could ruin the whole dish.
So mutations, they can cause disease.
Potentially, yeah.
But they're also kind of what drives evolution, right?
Absolutely. That's the source of variation.
So it's a double-edged sword.
In a way, yeah, it's a constant interplay between those genetic variations and the pressures of the environment.
That makes sense.
Yeah.
This all brings us back to that DNA profiling, like how scientists use those variations to identify people.
It's amazing, isn't it?
Yeah, and like trace their ancestry. So how is it being used today? Like, what are some examples?
Okay, so one big one is forensics.
Yeah.
They use DNA evidence from crime scenes.
Right.
Compare it to suspect DNA.
Like a genetic fingerprint.
It is. It can link them to the crime or exonerate them.
And paternity testing too, right?
A huge one. Yeah. Provides conclusive answers, sometimes even brings families back together.
That's amazing.
It's also crucial in medicine for diagnosing genetic disorders.
Right. You can see if there's a mutation causing a disease.
Exactly.
Or even like if someone's at risk for something.
Exactly. And it's not just diagnosing. It's also being used to personalize treatments.
Oh.
Tailoring them to an individual's genetic makeup.
That's amazing.
It's called personalized medicine.
So instead of like one size fits all.
Right. It's getting very specific.
You can see how they'll respond to a drug or therapy. That's incredible.
And then you've got all those genealogy websites.
Right.
People are using DNA profiling to trace their ancestry, uncover family secrets, connect with long-lost relatives.
So it's connecting us to our past and our future.
It is. It's pretty powerful.
But this is powerful technology, right?
It is.
There's like ethical considerations.
Oh, for sure. Privacy is a big one. As those genetic databases grow, we have to make sure that information's protected and used responsibly.
Yeah, for sure.
Absolutely.
Well, this has been fascinating.
It has.
We've covered so much.
Yeah.
The steps of protein synthesis, DNA profiling, and I remember you mentioned something earlier about DNA existing outside the nucleus.
Oh, yeah.
I definitely want to hear more about that. Yeah, so like, where else is it? It's kind of mind-blowing, right? Like we think of DNA and we think of the nucleus.
Yeah, it's not the only place. You got to remember, cells are complicated.
Yeah.
Lots of stuff going on. And inside each cell, these tiny powerhouses.
The mitochondria.
Mitochondria, exactly.
Yeah. Those are like the energy factories, right?
Yeah, the cell's power plants pumping out ATP, which is like the fuel.
No, it's like that's what powers everything we do.
It really is, yeah. And here's the kicker. Mitochondria, they have their own DNA.
What?
Separate from the DNA in the nucleus.
So like a whole other set of instructions.
Yeah, we call it mitochondrial DNA or mtDNA.
Why? Why would they need their own DNA?
Well, that's a bit of an evolutionary mystery. But the leading theory is that mitochondria, way back when, they were actually free-living bacteria.
Really?
Yeah, billions of years ago.
Wow.
And they got engulfed by early eukaryotic cells.
Like a cellular merger.
Exactly. And over time, they developed this symbiotic relationship. Mitochondria became a permanent resident.
So they brought their DNA with them.
Exactly. Never left.
So our cells are like ancient ecosystems.
It's like a history lesson in every cell.
That is so wild. Okay, so mtDNA, how is it different from like regular DNA?
Well, for one, it's shaped differently. mtDNA is circular, whereas the DNA in the nucleus is linear. It's also inherited solely from the mother.
Only from the mother?
Yeah.
How does that work?
So when a sperm fertilizes an egg, typically the sperm's mitochondria, they get destroyed.
Okay.
So only the egg's mitochondria are passed on.
So it's like a direct line.
Direct maternal line, yeah.
From our mothers or grandmothers going way back.
Exactly. And mtDNA, it mutates at a fairly predictable rate.
Okay.
So scientists can use it to trace human migration patterns.
Oh, wow.
Even estimate when different populations diverged, like a genetic clock.
So it's like a window into human history.
Yeah, it's pretty amazing.
You said it also plays a role in like energy production, though.
Right. So remember, mitochondria, they're the ATP factories.
Yeah.
The genes in mtDNA, they code for some of the proteins involved in that energy production process.
So if there's mutations in the mtDNA,
Uh-oh.
that could be a problem.
Yeah, it could affect energy production, and that's why mutations in mtDNA can lead to a bunch of health problems, often affecting organs that need a lot of energy, like the brain, the heart, muscles.
Wow, so these little tiny structures.
Tiny but mighty.
They're like so powerful.
They really are, yeah.
This has been amazing.
It has. It's been fun.
We went way beyond the basics today, talking about DNA, how it makes proteins, DNA profiling, even mtDNA.
Yeah, we covered a lot of ground.
Big thanks to DNA, The Code of Life by Mr. Chi for like helping us understand all this crazy stuff.
Yeah, it's a great resource.
And to all of you listening, we encourage you to, check out the book, keep learning, and until next time, keep those brains buzzing.